In today's video, we will be exploring the ethics of gene editing. How far is too far? First, we will be beginning by looking at a history of gene editing and its precursor, genetic engineering. Then we will explore what exactly gene editing is. Then we will follow what is known as the ethics protocol to see if gene editing is something that we should do. And finally, I will be giving my recommendations for how we should move forward in our study and application of gene editing. First, let's begin with a quick history of genetic engineering. So genetic engineering was developed in 1973, and shortly after its discovery, there was a moratorium staged for genetic engineering experiments, during which time scientists, lawyers, and government officials held a 3-day conference to set regulations for genetic engineering research. Then, some years pass, and more genetic engineering advancements are made, and somewhere here around 1985, the US government releases a detailed guideline surrounding the oversight of genetic engineering research, involving many government agencies to regulate the process. Throughout this whole process, the general public has always been quite wary of developments in genetic engineering because they could have potentially very serious social consequences. So in the context of genetic engineering, the human genome is comprised of many chromosomes, and chromosomes are just compact versions of DNA. So this is a strand of DNA. And if you zoom in on a section of DNA, you'll find that there's many genes. So here there are 2 genes in this section of DNA. And in genetic engineering, we can, for example, add another gene. So originally this organism does not have gene 3, but we can introduce gene 3. Say gene 3 comes from a different organism, and gene 3 will alter the properties of this organism. So this is genetic engineering. Gene editing, on the other hand, using CRISPR, what it can do is that it can take a gene, and this is the zoomed in DNA sequence of a gene, and DNA is made up of many nucleotides, and each ladder rung that you see here is one nucleotide. And what CRISPR gene editing can do is that it can edit any of the nucleotides that you want. So it can change it to a new nucleotide. It can change several nucleotides even. And by doing this, we've modified gene 1 into this new version of gene 1, which has never existed in this organism or perhaps in any other organism before. So we've created a completely new gene. And this new gene could potentially have properties that we want, or it could have properties we don't want. And as of now, it's difficult to determine exactly how this new gene will behave. So a brief history of CRISPR gene editing starts in 2012 when the mechanism of doing this gene editing is discovered in bacteria. Then a year later, we've adapted it to be used in eukaryotes, which are things like animals and humans, plants, so higher order organisms. And then just 2 years later, CRISPR is used to edit a human embryo. And even though this human embryo is a non-viable embryo, meaning that it cannot be implanted to lead to a pregnancy, but this is still a huge step. And finally, 3 years later, in 2018, the first human trials for CRISPR therapies to treat human diseases are approved. So as you can see, this is a much quicker timeline than what we saw previously with genetic engineering. And a concern is that because it has all developed so fast, government rules and regulations have not been able to keep up with the rate of gene editing scientific advancements. And as of now, there are many guidelines per se on what sorts of gene editing can be done, but these are just guidelines. They're not strict rules or laws or anything. And in fact, the actual rules are still pretty blurry. Now let's take a deeper look into what kinds of gene editing there are. So there are two kinds of gene editing, namely somatic and germline gene editing. So somatic gene editing is changing the DNA of someone's somatic cells, and these are just cells in the organs in the body that are not the reproductive cells. And the consequences of this is that it will change this one person's genes, but it cannot be passed down to future generations. However, for germline gene editing, this is changing the DNA of reproductive cells or embryos. So because it's reproductive cells and embryos, this can be passed down to offspring and it will remain in the population's gene pool. With these two types of gene editing, let's look at them one at a time and see what each could possibly lead to. So let's start with germline gene editing. So germline gene editing can do a lot of pretty crazy things. So for example, it can change human evolution because we're altering the gene pool. The gene pool is the collection of genes that belong to a certain species. So by changing it, we're changing the entire future of this species. And although we could potentially have benefits such as, say, we eradicate a certain genetic disease forever, these benefits are far outweighed by the negative consequences that could potentially result when we modify the gene pool permanently, because we have no idea what the long-term consequences of changing any one gene is. So to know that this gene has been changed permanently forever, that's a really big risk to take. And second, the issue of designer babies has long been a scary thought, because now that we're modifying the genes of embryos, we can construct embryos by choice, essentially. And this will lead to a lot of social disasters with parents trying to create the best baby possible. So as a result, germline gene editing is currently still very dangerous, and there are not that many instances where it's absolutely necessary. So as a result, the current guidelines on germline gene editing is that any editing done on embryos, the embryos cannot be implanted and cannot lead to pregnancies. And in the US, the NIH will not fund any germline gene editing research. And especially in recent years, there was a scandal when a Chinese scientist, He Jiankui, edited embryos and later implanted them into a woman, and actually these babies were brought to term. And this caused a huge backlash within the scientific community of He's actions. And this led to a moratorium on research into germline gene editing. Let's next talk about the safer form of gene editing, which is somatic gene editing. So what could we potentially do with this? Well, the very clear benefit of somatic gene editing is that it can lead to cures for genetic disorders. Genetic disorders are rare individually, but collectively, amongst all the genetic disorders, over 200 million people in the world have them, and that's around 3% of the world population. So it's quite a good amount of people with genetic disorders, and their lives could really be improved with this technology. But some of the downsides of somatic gene editing include, first of all, there may be errors in the editing technology because it's not 100% accurate. And especially given the current state of the technology, it is still quite risky to be doing this editing in humans. Second, as I said previously, the long-term effects of changing a gene is unknown because every gene has multiple functions. So it could cause a genetic disorder, but it can also be contributing to other things. So when you change it, you're not sure of all the possible effects that it can have. And third, Gene editing technology may easily be abused if wealthy people want to use gene editing to have enhancements. For example, athletes might want to enhance their physical capabilities, and other people might want to enhance their mental capabilities, and gene editing technology could be exploited for this purpose. So now that we've seen some of the potential consequences of somatic gene editing, let's explore somatic gene editing further and think about who the stakeholders are in this problem. So a stakeholder is potentially the wealthy people that I just talked about, who they have the means to use this gene editing technology for good or for bad. We also have got the biotech companies who mass produce this technology and can administer it to patients and can make money off of this. We've got the scientists who are doing the research into exactly how to do the gene editing, which genes should be edited, and how to edit genes to treat certain disorders. And then we have governments who need to oversee all of this and set rules and regulations onto how this gene editing technology can be used. And then we have the patients, people with genetic disorders who might really benefit from this, as well as the actual patients who are receiving the gene editing therapy. Now that we've considered these stakeholders, we need to consider what their values are. To identify values, we will consider 3 different categories of values, namely outcome, process, and structure. So outcome means what are the consequences of pursuing gene editing research and applications on these groups of people. So for people with genetic disorders, the gene editing technology can really save their lives. For scientists and biotech companies, they can make a lot of money using gene editing technologies. And finally, the wealthy people may abuse the technology to get what they want at the expense of the rest of society. Then let's consider the process. Process refers to who has a say in how the gene editing research, development, applications are done. Are people treated fairly when these decisions are being made? Do they have a say in, for example, what gene edits are being researched, as well as if certain edits can be done in humans? For example, scientists from wealthier nations may not want to research genes that affect diseases that are more prevalent mainly in poorer populations. A gene editing technology needs to be safe enough before it can be applied to humans, and patients really need to have a say in this. And finally, let's consider the structure, which means how are these resources and this gene editing technology, how is it distributed amongst people? Is it distributed in a fair way? Does it further exacerbate existing social inequalities? And here it's very important to note that not all patients will have equal access to the gene editing technology because it will be quite expensive, at least when it first comes out. And perhaps some patients who really need it the most might not be able to afford it. So now that we've thought about these values, let's consider the important choices that we need to make when we are thinking about how to move forward with somatic gene editing research and applications. And let's think about how these choices play into those values that we just talked about. So a first choice is, how should research into somatic gene editing continue? Who should be researching it? And what sorts of genes or genetic disorders should they research? Then we should also consider, how do we decide which gene edits are allowed in which populations? Because we cannot just have, anyone who wants their gene edited to get it edited, right? We need to decide who really needs it and which gene edits are safe enough to be done in people. Relatedly, our next question is, when should a certain gene edit be allowed to be done on patients, and who oversees this editing process? So how do we know this editing technology is safe enough to be applied in humans? And how do we ensure accountability that the people doing the editing are doing what they said they would do, as well as how do we make sure that patients really have consent in the process. And finally, we need to consider who will cover the costs of gene editing therapy for patients. You can imagine that patients who really need the gene editing therapy are patients that already have genetic disorders, and they probably have already spent a lot of money looking for treatments and finding ways to live with their disorder such that they might not have a lot of money left for the gene editing therapy. So we need to consider how they will be able to get the therapy. So thinking about all of these choices, I have some recommendations for what I think is the best approach moving forward. So first, I would strongly advocate for more research on how to do somatic gene editing more effectively, because right now one of the main barriers for its use is that the technique is not mature enough and not accurate enough for it to be done in humans. So we need more research on how to do it more accurately and safely. Then, we should have scientists of diverse backgrounds study the genes involved in a wide variety of genetic diseases. Like I alluded to previously, we don't want all scientists focusing on genetic disorders that primarily affect wealthy nations while neglecting those that affect poorer nations, because that would exacerbate existing social inequalities. We want to make sure that there are diverse groups of scientists with diverse interests in different genetic disorders, and all of them should be given the resources to study which genes are involved in those disorders and how do we modify those genes in order to cure those disorders. Then we need to have a worldwide conference with government officials, scientists, lawyers, ethicists, activists, and all sorts of other people to discuss guidelines on things such as what genes can be edited in what ways in which kinds of people. For example, we might decide that we are only allowed to edit genes that directly cause a genetic disorder, and this has to be a serious, life-threatening genetic disorder. So it can't be any sort of, say, neurological impairment like ADHD, for example. Like, that will not qualify. We also cannot be editing genes that may contribute to something but not necessarily cause it, because that may potentially lead to people abusing this edit. For example, if we find that there's one gene that contributes to having ADHD and we edit that, then perhaps wealthy people who do not have ADHD might also want this gene edited so that they can enhance their cognitive function, and that would be using gene editing for enhancements. And this directly ties into in what way we can edit the gene. I propose that we only edit a gene that is mutated in a diseased person, and we edit it back to the normal version of the gene that is found in normal people. So therefore, if you're a normal person, there's nothing to edit for you because you already have the normal version. You cannot do anything to get an enhanced version, for instance. And we only allow these edits to be done in patients who clearly demonstrate that they have this genetic disorder to begin with. We also need to decide on what the approval process for allowing an edit to be done on patients is. In other words, who decides when this editing technology is safe enough for it to be done on patients? And I propose that if a scientist or a biotech company develops a way to edit a particular gene in a particular way, they need to have performed this edit on enough mice and other animals, and they need to have these animals live for a long enough period of time to be able to see that there are no unintended long-term side effects of this gene edit. And after robust evidence in animals, then they can apply for NIH approval of using this edit in humans. Finally, we need to discuss what the oversight of the actual editing process on a patient is. Which government offices will be responsible for overseeing this? And I propose that whoever is doing the editing on the patient must be supervised closely, And at every step of the editing process, they need to be submitting samples for verification that they indeed have edited this one gene in this one way that was previously consented to by the patient. So, for example, after they do their CRISPR editing, they should submit a tissue biopsy sample to the government agency, and the government agency, such as the NIH, can then sequence the sample to make sure that the edit that was consented to is there and no other alterations to the genome is there. This would prevent people trying to do under-the-table deals of editing other genes that may not have been approved for them to receive. Finally, I propose that the government should subsidize costs of gene editing therapy for patients who cannot afford it. And these would be patients in which we have sequenced their genome and it has shown that they have this particular gene that leads to this genetic disorder, and they do not have the resources, they do not have the money needed for the gene editing therapy. And in this case, the government should subsidize the costs of this. In this way, we make sure that the patients who need this technology the most can get it. Thank you so much for listening, and I hope you learned a lot about gene editing and what we should do about it moving forward.